Hello everybody, making a quick little video for project number 5. Now in this unit there's actually 2 projects. The first project you should already have done, and it's where you're researching some genetic disorders. Okay, that one I was looking at, I was like, should I make a little help video for this? I think you guys have got that down pretty simple. Basically you'll just research it, and in your PowerPoint you should have a slide for each of those items that it talks about there. Okay, so pretty simple. But this one right here, I wanted to spend some time on, make sure you understand, because it is going over a key and major concept that you need to understand. And so this one right here, I love talking about sickle cell anemia because it's so fascinating. It's a phenomena that I like to use all the time in my class. What's interesting about sickle cell anemia is it's a genetic mutation, and there's actually one genetic mutation that is quite prevalent in majority of people, and anyways, it's a very small mutation and change that occurs in their genotype that will cause these cells to turn sickle, which is kind of interesting. Okay, and so anyways, we'll talk about this a little bit, and we were going to look at, when we look at sickle cell anemia, we're going to see kind of how this happens. How does DNA cause for this phenotype? And this is an extreme phenotype. This is very painful for people that have it. It's a recessively inherited trait, and so people, if they are homozygous recessive, if they have 2 alleles for sickle cell, all their cells would hypothetically be sickle shaped. They have a hard time carrying oxygen. It does usually will lower the life expectancy of that individual. Okay. Another interesting about this is that looking at kind of the evolutionary standpoint of this, sickle cell anemia is found in high frequency in people of African descent, people that live in Africa as well. And so kind of interesting there. Answering the question comes about, well, why is that? It's a much higher prevalence than people in other parts of the world. Okay, and so we'll look at that a little bit as well. I'll actually talk about it a little bit here. Anyways, but what we're going to be doing is looking at how does DNA make a protein, and so to look at the impact that might have here. And so we'll be looking specifically at 2 processes: transcription and then translation. And so right here, it gives you actually the DNA sequence of a normal DNA strand and then the sickle mutated DNA strand. And so we'll be able to look and see, well, what was the mutation that happened here? Now to do that, I kind of wanted to model this for you, and so I'm going to make this a little bigger here for us. Okay, so right here we have our DNA. Okay, oops, a daisy. I need to paint. Okay. Let's see. Brushes. Okay. So we've got our DNA here. And I apologize, I'm not the best at drawing. Anyways, and when we look at this here, it might be helpful just to kind of see, okay, well, where did the mutation first occur? And so when we look right here, we have GGG, GGG, so we're looking good. CTT, C, uh-oh. So right here we have some mutations going on. Then it goes C, and then the rest are T's. Okay, so right here we had a mutation. Now these mutations can occur during DNA replication. Also, from environmental impacts could have had this as well. And what's so important here is that this must have happened, you guys, in either egg or sperm of an individual, and then was passed on to the next generation. Okay, and so when this happened, it must have then been passed on to generation 3, etc. And so mutations that happen in egg or sperm are kind of the mutations that will get inherited. If they don't happen in egg and sperm, say they happen in just like skin cells or some sort of body cell, they will disappear when that person passes, right? And because your DNA in your skin cells, you do not inherit, you do not pass on to your next gen. The only ones you do are right here, egg and sperm. Okay, so we've identified the mutation here, and so what we have to do is look at, well, what's happening? What impact does this even have? And so the first step that we have to take is transcription. Okay. Okay, so transcription. And simply what we got to do here is we got to make the next strand here. Okay, so right here we know GGG. So basically what we got to do is using this template here, we're going to make what we call mRNA. And mRNA is a type of RNA, and it is a messenger. It actually will take the copy of the DNA and be able to take it outside of the nucleus to be transcribed. Okay, and so how this kind of works in mRNA or in any RNA, it'll be the kind of the complementary to this. And so we would first have C, 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 G, A, G, A, 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 A. Okay, so there's our normal strand. I'm going to do it in green, the mutated strand. So we have, oops, C, that's a C, you guys. C, 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 G. Now the key difference here, you guys, make sure you understand, in RNA we don't have thymine. We actually have another base. It's called uracil. A, 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 A. Okay, so there we go. We've transcribed. We have an mRNA strand. We're looking at both ones. Now the next step here is what we call translation. Now translation happens on ribosomes. Okay, and basically what you'll do here with the ribosomes is we take this mRNA and it's going to read every 3 letters. Okay, so read every 3 letters here, and each 3 letters will code for a specific amino acid. And to show you guys kind of what that looks like here, let me pull it up. So let's see, codon wheel. And so you might be familiar with something like this, but these are different. This is a codon wheel that you can use. Just starting from the inside and moving out, you'll be able to see what amino acid it codes for. The cool thing is this assignment actually gives you the codons that will be used here. So I'm actually just going to snip this. And use it. Okay, so Control C, there we go. No, I don't want to save. And I'm just going to paste it here, right here to the side. Okay, so there we go. That we can use it to transcribe this. And I'm going to do this in a fun color. Let's do it in red. Okay, so let's transcribe the normal DNA first, and then we'll do the mutated DNA to see what difference there was. Okay, so CCC codes for what we call proline. Now I'm just going to write the first 3 letters here. So PRO, GAA, gluta, GLU, and then I'm going to put A here, glutamic acid. We have GAA, so again, same thing, glutamic acid, and then AAA is lysine. Okay, so this right here is our protein, is our amino acid strand, our polypeptide. Okay, that was produced. Now let's look at the difference here. So CCC, so we have proline. So far so good, right? GUU, valine, uh-oh. And then we have glutamic acid and lysine. So right here you can see there was a key difference that happened. Let's look at it brown. Valine versus glutamic acid. Now you guys, this is a mutation, and it's not a silent mutation. We would call this a, well, basically it just changed one amino acid. Okay, so we don't know if this is beneficial or harmful. We actually do know because it causes sickle cell anemia. Okay, but it's just due to 2 simple, what we call substitutions. Okay. So there were two substitutions that occurred here that altered the amino acids. The good thing is it didn't change any of the amino acids afterwards. It just changed the one. Okay. But that one mutation causes this impact of sickle cell anemia, which is crazy, right? And so what you guys will be doing is using this, you're actually going to do what I just did here. So tell me, what is the mRNA? So transcribe, translate the amino acids. Oh, one thing that I want to talk about here. Let me pull this back up. So it talks about right here anticodons on the tRNA. Okay, so right here, this is actually this is the codon on the RNA. What you have to do is make a tRNA. Okay, sorry, I had to pause there. I thought my kid was waking up. Anyways, all right. So, like I was talking about, you have your. Let me pull that back up here. So, during this process right here of translation, a tRNA will come in and bring in an amino acid, whether that be proline or glutamic acid or lysine or valine, whatever it is. But it will attach right here to a specific, and it'll have what we call the anticodon. Okay, and it'll just be complementary to this. So we'd have G, 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 C, U, etc. Okay, so this would be another tRNA that's going to bring in amino acid, and it would then bond these together at the ribosome to create this polypeptide chain. Okay, so anyways, right there, you'll be able to fill this puppy out using that, and then simply just answer these two questions, turn this puppy in. All right. So just look at that type of mutation that did occur there, and you guys should be good to go. Anyways, if you have any questions on this one, let me know, but I hope this was a clarification on how to do this project here so you guys can just ace it.